I am Parmanand, PhD student at Amity Institute of Biotechnology, Amity University, Haryana, Gurgaon, India. I am pleased to present my research work at South Asian Network on Bioinformatics Conference. Today, I am presenting my work entitled as Targeted Warfare, Fast Therapy Against Proteus mirabilis strain indica. Proteus mirabilis is an important opportunistic pathogen and a major cause of catheter-associated urinary tract infection. The increasing emergence of multi-drug resistant strain along with their ability to form robust biofilm has made the treatment of these infections increasingly challenging. Therefore, there is an urgent need to explore alternative antimicrobial strategy. The objective of this study was to isolate and genomically characterize a new novel lytic bacteriophage capable of targeting multi-drug resistant Proteus mirabilis. For this purpose, seaway samples were collected and screened for bacteriophage using multi-drug resistant clinical isolate of Proteus mirabilis as a host. Following that enrichment and multiple round of plague purification, we successfully isolated a novel lytic bacteriophage which was designated as Proteus phage Ramarti 1324. The phenotypic characterization of this bacteriophage was characterized by using transmission electron microscopy, and it was showing that this particular phage have icosahedral head and short non-contractile tail. This is the characteristic feature of the podogiridae viruses. So this bacteriophage is falling under the family of podogiridae. And to investigate the genomic composition of this bacteriophage, the whole genome sequencing was performed and this genomic data was analyzed, and it was characterized that this bacteriophage have 146 protein coding genes along with 4 tRNA. Among these 146 protein coding genes, this particular bacteriophage have two different endolysins, that is making this bacteriophage to be efficient against killing the Proteus mirabilis. And also this bacteriophage genome has free has no any virulence factor genes and no any antimicrobial resistance genes, so making this phage as a good candidate for the antimicrobial agent. Also, the comparative genomic analysis was performed against the 51 available protease infecting bacteriophages in the NCBI database. So the genomic data from for the Proteus mirabilis infecting bacteriophages was taken from NCBI, and this is a total around 51 bacteriophages. And the genome was compared to understand the real genomic relatedness of this bacteriophage with the other available bacteriophage genomes. And it was found that this particular bacteriophage named as Proteus phage mirabilis 1324 is closely related to Proteus phage privateer and also contains some unique genomic region that is making it novel candidate against the Proteus mirabilis. Furthermore, the phage exhibited significant activity against biofilm also. Against, again, this comparative genomic analysis was performed and through the protein clustering and also the genomic analysis, and the unique genomic feature of this protease phage was characterized by protein clustering.